So, welcome to our participants to our 12th ISG webinar. You're heartily welcome. If you missed something, you know that you can see all of the webinars on demand later on. Latest on Tuesday, they should be available on our website. I think you also know that when you are registered and you need the points, the CME points, then you will receive a questionnaire evaluation folder. Please send that back, and then you will get all the points you deserve, and together with some kind of diploma. And may I also ask you to put your questions, if you have some, to the question and answer folder here inside, not to the chat. The chat is really limited to the faculty who gives presentations. So with that, first announcements, Pika. You may start with that one. Pika from Copenhagen will start the whole webinar with Epidemiology of Toxoplasma Gondii infections. Enjoy.
So thank you again for the invitation. I'm very happy to be part of today. My name is Pekka Jokelainen. I am based at State and Serum Institute in Copenhagen, Denmark. This is a public health institute that also has veterinary preparedness functions, and we have the diagnostic reference lab for parasitic diseases, including toxoplasmosis. And my background is a lot in One Health work, so it is exactly thinking about zoonotic parasites from a little bit bigger picture. So with my talk, I hope to bring you all to think this way. So we will later focus in the eyes, but first to talk about Toxoplasma gondii as a parasite and the epidemiology of the infections. When I talk about these parasites, it probably comes true that it is my favorite parasite, and I find it extremely fascinating. I have been myself doing research on it for about 15 years, and there's just so much to learn about this parasite. First, I would like to emphasize that when we talk about epidemiology of Toxoplasma gondii infections, it's important to think when we talk about the infections and when we talk about the clinical disease toxoplasmosis. Toxoplasma gondii is really a parasite that is, I would say it's a good parasite. It doesn't harm the host too much usually, and this enables it to continue and find new hosts and live long and happily in a host. But indeed, mainly we have information of how common the infection is, and on the other side, we may have information how common a clinical disease is. When we talk about the infections, we often use the word asymptomatic if there's no specific clinical manifestation. I would a little bit challenge this, especially for people who are unable or not really good in expressing themselves, also small children, for example, and also the animal hosts. So there I would say, like, maybe we sometimes should talk about subclinical infections rather than asymptomatic. There might be symptoms that are just not attributed to Toxoplasma gondii. And as you already well know, there are many manifestations of the clinical disease, including ocular toxoplasmosis, and overall this infection leading to the disease can be even fatal in both humans and in animal hosts. This figure here is not the eyes, because we will see a lot of pictures from the eye later. This is other tissues, liver, lung, and central nervous system from domestic cats. So even cats that are the definitive host can even die from toxoplasmosis. So how common is Toxoplasma gondii infection? I would say it's too common. It's really prevalent different places in the world. This is currently unpublished data from Denmark from 1994 to 2010. This is seroprevalence of Toxoplasma gondii in Denmark. This is samples sent to our laboratory, so a biased sample. There's referral bias, certainly. So it's not, in that sense, representative, but clearly shows that seroprevalence increases with age in humans. So there is the age in years in x-axis and proportion IgG positives on the y-axis. And what is important to know that it starts quite low. So the congenital infection is a small part of the whole pack of infections we have. And it seems to go up throughout the lifetime. So it is really typically an acquired infection at some points of the life. What is interesting that this figure is very similar. Here we have genders, male and female males are the blue one and females are the red one. And why the red go a little bit lower is again a referral bias, because those are largely women that have been tested during or before pregnancy. So that's why it's not totally the same. But what is interesting that it's very similar to other hosts. So here I have two graphs of seroprevalence by age in years in domestic cats. The one above is from Finland, and the below one is from a Baltic country, Estonia. Both are work of my PhD student, Kert Must. Really similar pictures. So it is an acquired infection in most host species. And also these numbers, if you look at how large a proportion of cats, it's about half of the cats are seropositive, but most have no clinical disease. And same is in humans, that the clinical disease is a smaller part than the seroprevalence or the infection prevalence. When we talk about Toxoplasma and epidemiology, we always need to go back somehow to life cycle or transmission routes. And I took this illustration here that I have been adding a little bit around. Key point to remember is that Toxoplasma has 3 infective stages. So the oocysts, the environmental form, is one, and then zagozoids and paradozoids in the tissue cysts are the tissue forms. And what is quite striking, actually, that even though we know a lot about Toxoplasma to the smallest detail, and we have investigated both the parasites and the diseases it causes, we don't really know where humans get the infections. So the knowledge on what is the proportional importance of these different routes to humans is actually largely unknown. And currently we are having in Europe a large project working on this, exactly called toxosources, where we try to use different approaches to get some answers to what is the proportional contribution of different sources. Very often we talk about Toxoplasma as a foodborne or waterborne infection. At the same time, that is also kind of, I would say, rather thinking about the foodborne actually also includes the oocysts. So there is a lot of food we eat where it's not the tissue cyst, it's actually oocysts that may have contaminated the food. And more focus has been on the oocysts as a possible source. And there I would like to emphasize that they are impressive in how resistant they are in the environment. This is just one table from Duby's article, or where there's an arrow pointing to one number. This was Toxoplasma gondii sporulated oocyst kept in +4 Celsius degrees in water for more than 4 years, and they were still infective. So as soon as the oocysts are out there in the environment, then it's a long-term contamination of the environment, and it can contaminate water or food or gardens or whatever we are, children's play yards. And thus I would like to everybody to, from now on, to think about these life cycle illustrations where we have the arrows, that how could we better stop this, because it's really the oocyst contamination is a long-term challenge and problem. So if you think about usually how we try to stop human Toxoplasma gondii infections, we focus on the really last arrows going to the humans or going to the unborn child there. And then we could think a little bit kind of One Health thinking, where do these parasites come to the food or water that is the immediate source? And for example, those animals that are raised or hunted for human consumption, if we could lower the prevalence in them, that would help a lot. Where do they get the infection? They are usually herbivores, so they typically get their infection from the oocysts. So where do the oocysts come from? There's the cats, but actually cats, if the cat itself doesn't get the infection, it will not shed the oocyst and it will not contribute to the success of this parasite. And we have done several studies where we looked at domestic cats that are kept as pets, and major risk factor for the cats to get this infection is that if the owner is giving raw meat to the cats, and raw meat that then is from an animal that has the infection. So there's like a circle that I actually feel like we humans are currently helping, and we could maybe limit that better. So basically, no raw meat to the cats, and cat feces should be collected and disposed of properly so that they don't contaminate the environment. And we should think about the food chain all the way. Where can the parasites enter it? And then also, of course, remember the last arrows just before the humans. So there's a lot to do in this. Another point to think about the epidemiology of Toxoplasma, it is really global. It's actually quite successful globally. This colorful picture is from our paper where we looked at clinical samples that were tested for Toxoplasma Gondii DNA with PCR methods in our laboratory in Denmark, where we in Europe, we expect mainly to have a genotype 2 of Toxoplasma. And that would be the one in a bright green. That is the group there. And in this figure, the little triangles, those are the ones that were from our samples. So samples sent to our lab in Denmark, and they were not only in that corner. We really saw a higher variation, genetic variation than we expected in this quite small study. This included actually samples from ocular toxoplasmosis. And this was kind of eye-opening also to realize that there is, there's a lot to think about it, and we really need data globally on the epidemiology of Toxoplasma. Where is it? What are the strains? How is it circulating? And we should remember that we are living in a really globalized world. So we were able to find strains that we didn't expect in a higher proportion also. And as my really last slide to open this discussion is that I've looked at this tissue cyst of Toxoplasma gondii. I really was thinking it was like the full moon. And this is really the thing that for both understanding Toxoplasma, we need international collaborations, and also to remember it that it is basically everywhere on this planet, and it's doing well, and it's going strong. So we need to be doing something to lower the prevalence along the whole chain. Thank you.
Pika, thanks so much for this very lovely introduction to the topic. Next, Alejandra de la Torre. She will talk about the pathophysiological aspects of ocular toxoplasmosis, especially the host-parasite interaction. Alejandra?
Yes. Good morning. Are you hearing me?
Perfect.
Okay, perfect. So good morning, good afternoon, and good night to all the connected colleagues worldwide. I want to thank Dr. Manfred Zierhut for inviting me to participate in this webinar to talk about pathophysiological aspects of ocular toxoplasmosis host-parasite interaction. While Toxoplasma was discovered more than 100 years ago, recently, we are beginning to understand the importance of the host-parasite interaction on the parasite growth, radiculocyte formation, immunization, and virulence, particularly in the peculiar intraocular environment. Today, I will summarize the interplay between T. gondii and its hosts and the possible implications on OT's clinical manifestations and outcomes. Different host and parasite factors have been identified as disease modifiers. What factors do we have to consider in the interplay between the host and the parasite? Host factors include genetic predisposition, immune status, ocular immune privilege, age, and pregnancy. The parasite factors include geographical distribution, stage, strain, inoculum size, and the source of this inoculum. Let's start with the parasite. We already know that the parasite population of T. gondii has a biphasic geographical pattern. One in the northern hemisphere, where few highly clonal and abundant lineages predominate, mainly type 2, which are less virulent. And the other one in the southern hemisphere, characterized by a diverse assemblage of less common genotypes with more significant evidence of recombination, mostly type 1, 3, and atypical strains, which are more virulent. When studying the clinical consequences of the diversity of Toxoplasma gondii strains, we can see that European patients present different clinical pictures when compared with South American ones. Not only in congenital toxoplasmosis, but also in ocular toxoplasmosis in adults. That's how South American patients infected by less virulent strains, more virulent strains, mostly atypical strains, present severe ocular toxoplasmosis and more extensive lesions and also more inflammatory lesions and a Th2 deviated immune response. On the other hand, European patients infected with less virulent strains, mostly type 2 strains, present mild ocular toxoplasmosis, less inflammation, smaller lesions, and a Th1 and Th17 deviated immune response. When correlating clinical characteristics of ocular toxoplasmosis patients with the infecting strains, and with the local immune response and genotyping and serotyping, we can see that the European patients present different clinical picture and also different intraocular immune response when compared with South American patients. European patients infected by less virulent strains, mostly type 2 strains, present a deviated intraocular immune response to Th1 and to Th17 with higher levels of interferon gamma and interleukin 17A, leading to a limitation of the parasite proliferation because those are pro-inflammatory factors and protective factors, and leading to a moderate retinal pathology. While South American patients infected by more virulent strains present an intraocular response deviated not to Th1 or to Th17, but to Th2. With lower levels of interferon gamma and interleukin 17, leading to an uncontrolled parasite proliferation and to a severe retinal pathology. The local intraocular immune response is deviated according to the infecting strain, leading to an ocular cytokinome different between European patients and South American patients, and this cytokinome is linked to clinical characteristics in ocular toxoplasmosis. The Th2 response is related to more severe clinical characteristics in patients infected with type 2 and atypical strains, and these patients have a higher parasitic load. Severe South American ocular toxoplasmosis is associated with a decrease in interferon gamma and interleukin 17, and an increase in IL-13 and IL-6. We also found that IL-5 and VEGF were associated with recurrences. This is about intraocular immune response. What about peripheral immune response? The peripheral immune response against virulence factors such as the surface protein P30 and rupture factors such as ROP18 is also shifted towards Th2 in South American patients. We know that in murine models and in vitro models, Toxoplasma gondii modulates the host by specific proteins on the parasite surface, also in micronems, ruptures, and dense granules. These are some of the proteins associated with the Toxoplasma gondii invasion process. One of these proteins are related to the attachment, and other ones are related to the penetration, and another are associated with the internalization. We have been studying for the first time in humans some of these proteins, such as SAC1, located in the surface of the parasite, specifically P30 protein. We have also been studying rupture proteins associated with the penetration, such as ROP18 and ROP16 proteins. And we found that patients infected with strains with ROP18 virulent alleles present more severe inflammatory response. And most of the ROP16 nucleotide sequences from Colombian patients with OT belong to the group of mouse virulent strains. Patients with more severe ocular involvement infected by strains with ROP18 and ROP16 virulent alleles present more severe inflammation. These strains probably came from water rather than from meat. One example of the clinical consequences of the infection with atypical strains with ROP18 virulent alleles are these immunocompetent patients with no history of exposure to jungle environment that present severe acute multisystemic failure with an extensive intraocular involvement. We already talked about parasite factors. Now we will talk about host factors. One of the most important factors is genetic predisposition, because genetic polymorphisms are associated to susceptibility to develop OT. Another important factor is immune status, because immune system maturation and immunosuppression are related to infection control effectiveness. Another important factor is ocular immune privilege, because immunological intraocular environment restrains the inflammatory responses. Another factor is the age, because active OT is more prevalent in younger individuals, but patients with recent seroconversion are significantly older. And extremes of age are risk factors for increased severity. Pregnancy is also important because primary acquired T. Gondii infection during gestation may lead to a maternal-fetal transmission, and a distortion of the immunological equilibrium could interfere with parasite control. Here you can see some of the genetic polymorphisms related to toxoplasmosis presentations. Some of these polymorphisms are described in Brazil, another in the United States, other in Australia, Poland, Colombia, and the United Kingdom. We cannot consider parasite and host factors separately, since they have an interdependent relationship. The parasite mechanisms of intraocular invasion and the subsequent immune response are crucial. Although the final target of the infection is the eye, the parasite enters through oral ingestion, invades the epithelial cells of the gut and the lamina propria infects the innate immune cells, reaching the bloodstream and crossing the blood-retinal barrier through different mechanisms, subsequently transforming into a cyst and establishing latency. Invasion of the host cells is mainly an active parasite-driven process that depends on the infection of various and in the interaction of various cell surface ligands. The immune microenvironment of the eye, designed to reduce inflammation and preserve transparency, may have a limited capacity to clear Toxoplasma gondii infections. Once the parasite reaches the eye, crossing epithelial and endothelial barriers through different mechanisms, the parasite can stay into the eye, leading to different immune response according to the type of infecting strains and leading to a different retinal pathologist. Other determinants of the disease are social and cultural factors such as eating behaviors and environmental factors such as mean rainfall. In conclusion, host-parasite interaction in OT is a complex and multifactorial relationship, with the parasite always on the driving edge of the game. None of the factors alone can fully explain OT pathophysiological phenomenon. They could be seen as an integral relationship. There are still multiple incompletely understood fields to be investigated. Future research would permit further insights into the immune biology of the parasite and recognition of the host-parasite interplay to improve diagnosis, outcomes, and treatment. Thank you very much.
Thanks so much, Alejandra. Beautiful presentation. So now we have the situation that I think that José Montoya is still not with us. We have to skip that at the end. Hopefully, it's a time problem. It's 4 o'clock there on the West Coast, so we have to wait a little bit. Probably he joins us later. And I would like Justus Garvik from Bern, from Switzerland, to start his presentation about diagnostics for ocular toxoplasmosis, clinical diagnosis, serological testing, and the role of AC taps, and finally also images. What a big topic.
Thanks, Manfred, for the invitation. Dear colleagues and friends, after the fantastic talks before mine, I come now down to clinical aspects of diagnosis, serological testing, and new imaging modalities. My disclosures, just to come back to the first talk of PICA, 30% of the world population are Toxoplasma antibody positive. And the seroprevalence of Toxoplasma antibodies in US and Europe is 15%, while in the Southern American countries, it may be 65 to 80%. This indicates that every third patient in the world, on average, harbors toxo antibodies. So if you do serology testing, you may exclude infection if you have no toxo antibodies. But if you do find toxo antibodies, this is not very much contributing to the differential diagnosis. And this is much more the case in European countries, where more than half of the patients are toxo antibody negative, compared to Southern America, where 20% of the patients are toxo antibody negative. In Europe, it has been calculated that 3 million patients have active ocular toxoplasmosis or related scars, and the majority of these are unrecognized asymptomatic scars. If you look from the aspect of uveitis, then you see that every 12 patients with uveitis has ocular toxoplasmosis. So 1 out of 12 of your uveitis patients may be a toxo case, and 2/3 of them have active disease, while even in active uveitis, you find sometimes cases with inactive scars typical for ocular toxo, which has never been reported before. As an ophthalmologist, you do not look whether a disease is acquired or congenital in origin, because clinically it's very difficult to differentiate. if you look at this work from 2008 with more than 400 patients, 2/3 of the patients had no clear determination of the source of their disease. It may be that the majority was acquired and the minority congenital, but generally speaking, of the diagnosed cases, 2/3 are acquired and 1/3 is congenital. Congenital is not the majority, but the minority of cases. Whereas in the bilateral cases, congenital disease is clearly prevalent. So if you have a case with unilateral disease and an active lesion, a large active lesion in an elder person, it's likely to be acquired, while a young person with a bilateral disease, it's likely to be congenital in origin. The problem why we cannot differentiate clinically is also because congenital toxoplasmosis is present since birth, but it may manifest with the first ocular episode as late as 17 to 20 years of age. So you cannot say a patient who is 20 years old must have acquired disease. It could still be congenital disease. And now we come to the clinical imaging. You see here a case, a young male with claiming progressive floaters, a little bit of nebula, but a full visual acuity. And this is typically in the majority of cases which have acquired disease. On the left hand, you see that the lesion is at the inferior vessel arcade. On the right hand side, you see the diffuse infiltration in the OCT, and also you see the preretinal infiltrate. These hyperreflective spots resemble vitreal infiltration. So this case clearly has ocular toxo. It's very typical because his lesion starts in the retina and not in the choroid, meaning it's first a retinal lesion and then a choroidal lesion, giving it the term retinochoroidal and not chorioretinal lesion. And on the right-hand side of the OCT, you also see the pigmented epithelial involvement, which is very typical of active ocular toxoplasmosis lesion. Generally, visual acuity is a problem in the minority of cases, while the majority of cases have early and persisting visual field defects. This may well be explained by the fact that toxo affects the inner retinal layers, and that means the nerve fiber layer. Affection of the nerve fiber layer means large visual field defects, while a chorioretinitis does affect the outer retina secondarily, and this means the nerve fiber layer is not effective. Because of that, we have the so-called post-horn defects in visual field, which can be very significant and are the reason for moderate to severe visual field impairment in more than half of the cases, while only 25% of the cases have a moderate to severe visual acuity affection. So if you want to know whether it's toxo or some other chorioretinal infectious disease, then you have to do visual field testing. If you have a so-called post-horn defect, which is not restricted, where the scotoma is not restricted to the lesion area, but much larger, then you likely have an ocular toxo, while a focal scotoma only is typical for tuberculosis or sarcoidosis. It could be borreliosis, or it could be in the area of viral retinal necrosis. So visual field may help, but visual acuity does not help in differentiating. Here you see a case where you might ask, what is it? You have a very active lesion, but you also have a huge vasculitis, which is not only in the lesion area, but if you look carefully, also in the nasal inferior branches of the central retinal vein and artery. So there's a predominant vasculitis, which is very diffuse, associated with a focal lesion. Clinically speaking, I should think, okay, this toxo, isn't this more likely to be tuberculosis or sarcoidosis, or even US, both or all of which are going along with vasculitis, much more than toxoplasmosis. Then you do the angiography. And what do you see in the angiogram? You see in the early frames, you see retarded perfusion of the affected vessel. and you see diffuse dye leakage in the affected area, but not so in the other areas where the chloralized plaques are visible, which indicates it's more or less a local phenomenon which diffused through the area, which is again in line of fitting to toxo, but you are never sure. And if you are not sure and you need to treat the patient because you have an extensive disease, you definitely would not want to treat with corticosteroids, which are clearly indicated in such a severe vasculitis, before you know it's not infectious in origin, or the infection is under control. And you look at the serum antibodies, then you see, okay, pretty high serum antibodies, not meaningful. You look into IgG avidity, which is again not meaningful. And then you look into the toxo antibodies, and then you see in the aqueous humor, you see no IgG. This indicates, okay, it must not be toxo. But if you detect a specific IgA in a significant level, which is in this ELISA that we use here, the Platelia toxo ELISA, it's 1.6 with a cutoff of 0.1, then you have to say clearly, okay, this is an early active toxoplasmosis, which is proven with the IgA in the absence of IgG. In this case, if you would do a second puncture 2 weeks later, you would likely detect IgG and also have a positive Goldman-Wittmer coefficient. And now I want to go through some cases which could or could not be toxoplasmosis. This is a 70-year-old female. She was referred for wet age-related macular degeneration on her better seeing eye. On the bottom, you see her worse seeing left eye with a huge macular scar associated with chronically active macular degeneration. And then you see the finding on her right eye, which has a significant cataract, but obviously also some vitreous infiltration. And you see this lesion. And if you look carefully, then you see the lesion is not in the macula, which means no, it is not age-related macular degeneration, as you in a first glance would think if you look at age. If you look carefully at the anterior segment then and detect mud and fat precipitate at the corneal endothelium, then you have to think about toxoplasmosis in the differential diagnosis, and it was confirmed in this lady indeed with a Goldman-Wittmer coefficient of more than 20. So she had an acquired atypical toxoplasmosis of the elderly, and you must not mismatch that with some age-related disease just only because she's old. Another old lady, she was referred for ocular toxo treatment with affection of her macula and severe vision loss. If you look at the image, you see clearly, okay, it's a retinal lesion. You cannot predict how much the choroid is involved, but it's surprisingly little vasculitis. It's surprisingly little vitreal infiltration, but there are retinal hemorrhages, which are not typical. The lesion size is not typical, and the hemorrhages are not typical for active toxo. In her case, we did multiplex PCR testing and found her to be CMV positive. She had a necrotizing osteomyelitis as the reason for her immunosuppression and the the CMV disease of her retina, which required a definitely different treatment than you would give for toxo. And you see another old lady, 81 years old, thought to be healthy, but she had fever of unknown origin for quite a time. And if you see bilateral lesions in the papillomacular bundle, in this case, with severe vision loss in her left eye and beginning vision loss in her right eye, then you have to come up with a diagnosis rather rapidly. In such a case, you definitely would want to use imaging technologies as well as antibody testing to make sure, antibody and PCR testing to make sure you do not miss a diagnosis or do come up with a diagnosis lately only in a case with impaired vision on the one eye and severe danger of severe vision loss in the other eye. This case from the clinical picture, it could be candida, it could be the tuberculosis, it could be toxoplasmosis, and it could be beginning viral retinitis, though bilateral symmetrically presenting bilateral viral retinitis is a rare case. In the OCT then, sorry, it's an old OCT, as you can see. You see the diffuse infiltration of the neuroretina, but you, in the upper image, you also see a thickened choroid, and you see again the pigment epithelial affection, which is typical for toxo, but not for CMV. In CMV, you would expect atrophy, which you did not see here. So from the imaging, you would tend to say, okay, it's probably toxo, but you also see in the upper image, it's very, very close to the foveal center, which is still nicely maintained. In that situation, you definitely would do an anterior chamber tap. And surprisingly, what did we detect? Extensively high IgG antibody levels in aqueous humor and serum, but also we detected very high IgM levels and very high IgA levels, which is indicative or is strong proof of acquired ocular toxoplasmosis despite her old age. And the Goldman-Wittmer coefficient was almost 120. So far to the smart running cases in Europe, this was definitely not such a smart running case. This would be a case as you would see it also in Southern America. We had not the opportunity to do strain typing in this case, but it may have been an atypical strain. Another case where you have to ask yourself, do I want or don't I want to do invasive diagnostics? A case with a little bit of symptoms, a little bit of visual field defect, a full visual acuity, and you see many peripheral scars on the left-hand image, an active lesion close to the optic nerve head, and associated with the scar at the nasal superior arterial vessel arc. In this case, we did do an anterior chamber TET because we wanted to confirm the diagnosis because of frequent recurrences and the consideration of a prophylactic regimen following an active treatment. And if you look at the IgG titers in serum, they're not very high. IgG avidity is low. So the systemic immune response in this case is not interesting. But if you look at the aqueous humor, then you see, okay, there's a very high or comparably high IgG antibody correlation in the aqueous humor. Avidity is high, as you would expect in a recurrent disease. But again, this case had a very high IgA level in the aqueous humor, but not so in serum. So if you do aqueous humor testing, as you've seen in 3 or 4 of our cases, IgA ELISA is very helpful beyond Goldman-Wittmer coefficient. PCR was not done in the case because of cost-saving aspects in a clinically pretty obvious diagnosis. Why could we miss a diagnosis? Why could be that the aqueous humor or serum antibodies are negative? If you look at a study that we have done a few years or some years ago, you see the serum at sampling and the serum 6 weeks later, they have the same levels. So meaning there's no change in antibody titers in serum in the majority of your ocular toxoplasmosis cases. But if you look at aqueous humor, do an aqueous humor test at diagnosis and 6 weeks later, then you see a clear increase in the local immune response. This indicates that if you do aqueous humor testing very early, which is 2 weeks or less after beginning of symptoms, then you may end up with negative antibody testings despite the diagnosis of toxoplasmosis because you have tested too early. If you test too early, then PCR is likely positive while antibody testing is likely negative. And another issue is that the serum antibody levels and the aqueous humor antibody levels in acquired disease are much higher than in congenital disease. Experimentally, we looked into that, sorry, and found that the antibody levels in the serum, but also in the eye, are not very high until 2 weeks after experimental infection, which again indicates 2 to 3 weeks are required for the antibody tests to get positive to confirm your diagnosis. If you puncture early, you may account negative results if you do not look for PCR. To summarize, the detection of intraocular antibody production using ELISA after 10 days has a sensitivity of only 63%. while PCR has a sensitivity of 55%, which is almost as high. If you look later, then the sensitivity of antibody tests will increase, while the sensitivity of combining PCR and antibody index adds to 80%. And here's a typical case where you would want to have the answer, is this toxo or isn't it? In my feeling, it's not a very typical case of ocular toxoplasmosis, but you have an old scar. If you look at the antibody levels in the serum and aqueous humor, you do not find very high levels, and you're not convinced it's a toxo. So low antibody levels can be associated with congenital disease. And the Goldman-Widmer coefficient was not indicated. In such a case, I would treat for toxo and any differential diagnosis, but I would do a second puncture 2 to 6 weeks later to confirm the diagnosis. If you want to know earlier what is going on, then you have to add immunoblotting and have to detect specific bands, as you can see in the aqueous humor panel on the left side of the immunoblot, with 2 very strong bands indicating aqueous humor antibodies specific to toxo. And this was the case in the patient that I showed before. He had specific antibodies, and this proved to be active toxoplasmotic disease, despite not being clinically very typical. If you have a case where you do not know what it is, you can also in the aqueous humor detect IgM bands which are not present in the serum, and again you can detect IgA bands. So if you have antibody testing with inconclusive results, it may be advisable to use toxo-immunoblotting, but not use IgG blotting only, because if you compare IgG bands in serum and aqueous humor on the right and on the left side on this image, then you miss the differences in IgA and IgM that are present. Here you have another case to show clinically what you could find, a case where there is no much activity, large scars, very typical of toxoplasmosis. The patient probably mainly noticed vitreal floaters, but the Goldman-Widmer coefficient is highly positive, IgA is highly positive. So obviously the antibody response does not closely correlate with disease activity. To summarize, the clinical diagnosis remains the diagnostic gold standard. Acquired disease compared to congenital toxoplasmosis is more frequent, more atypical presentations are found, and namely that in elderly persons. More severe inflammation but better visual acuity is the normality in acquired disease, because the lesions are not in the macula but outside the posterior pole, and also in acquired disease you have less recurrences. Visual field for the differential diagnosis, as I have shown to you, and functional damage because of the nerve fiber layer destruction may help you differentiate toxo from other sources of chorioretinitis. Imaging, namely OCT, may help in the differential diagnosis, proving that the disease started in the retina and not in the choroid. Laboratory confirmation of aqueous humor and serum antibodies and PCR testing in atypical cases is clearly justified and has a sensitivity, if combined, of 80% and a specificity of 98%. There are no qualitative difference in serology. If you use serology only without comparing to aqueous humor, this is not meaningful, except to exclude toxoplasmosis as and possibly underlying disease. Negative IgM is the normality in the vast majority of cases and does not exclude active ocular toxoplasmosis, because ocular toxoplasmosis is not usually arising during primary parasitemia. And this leads to an algorithm how to treat how to diagnose toxo. You have a typical clinical presentation, and you may, if you're not completely sure, test for serum antibodies. If these are positive, you do not do further testing. But if the serum antibodies are negative, then you have to go into the differential diagnosis of toxo. If you have an atypical clinical presentation and vision is not at risk, then you may look at the clinical course and do only serum antibody testing. But as soon as vision is at risk, then you have to use parallel diagnostics from aqueous and serum with quantification of antibodies, not only to toxo, but also to rubella, to be able to calculate the Goldman-Wittmer coefficient, and do PCR if the Goldman-Wittmer coefficient is below 2. If you do not want to do PCR or have no access to PCR, then you could do aqueous humor immunoblot testing, which is also highly informative in our hands. Here you see such an atypical case where you could not differentiate toxo from CMV because of the retinal images and the extensive retinal involvement. This was toxo, but we primarily thought it could as well be some lymphomatous infiltration or CMV disease. And here you see another atypical case, again, ocular toxo, where you would not want to come up with a diagnosis without antibody testing. So toxo is something you should consider, and with regard from Paul Klee from Bern, I end my talk here and hope you took something home with it.
Thanks much. Wonderful. Jose, can you see us?
Yes, hi, good morning. I'm here.
Good morning. Sorry about this time misunderstanding. What do you think? Would you like to start now?
Sure. Thank you.
Can you go back a little bit? We don't see your picture.
Okay, great.
Yeah, that's wonderful. Yes. Welcome.
Thank you.
It's very early in your...
For a very long time.
Sure. Hi, Karwek. Can you see my screen?
Can you start it? Yes, I think we can see it. Okay, so for all the participants, we now go back to the topic number 3. Jose Montoya will talk about the genetics of Toxoplasma gondii. Welcome, Jose.
Great. Thank you for having me here. I apologize for the misunderstanding. As you can see here in the announcement, says West Coast time 5 a.m. And it's close to 5 now, so I had prepared for this time. But anyway, so we are here. It's great to be among other speakers like Dr. Joke Leitner, De La Torre, Garweg, Belfort, and Jorge Gomez Marin. As you know, in our laboratory in Palo Alto, we name it after our founder and mentor, Dr. Jack Remington. He passed away this year, and we are mourning, but at the same time, making sure that we carry his legacy. This is our group. It was founded in 1962, and since then, uninterruptedly, we have carried the work that he left us to do. The parasite, as we all know, have 3 main forms that is used for transmission, given so the oocyst that is basically excreted in cats, and it has the tachyzoid form that is responsible for any manifestation. So when you see Dr. Garweg's presentation about how it causes inflammation and how it causes destruction in the retina, either as an association with acute disease or manifestation reactivation of chronic disease, this is the form that is triggering that inflammation, the tachyzoid, and the dormant form, the cyst form. So when you see these 3 forms, it's actually the same parasite, Toxoplasma gondii, and is basically spreading in nature primarily through the biological amplification. The biological amplification in the small intestine of cats. There is where there is recombination. There are forms that are generated, the macrogamete, which is the female form, and the microgamete, which is the male form. And there is an exchange of genetic material. So it is any member of the feline family where the parasite has the ability to exchange different genetic traits. And the more wild these cats are, the more extended territory they cover, the greater is their ability to exchange genetic material. So you can start to form the idea when you have cats in France that are domesticated, that are part of the usual urban cycle, is going to be very little chance for different strains to mate and to join. Whereas if you think about the vast Amazon areas in South America, where these big cats travel huge distances, that there will be more time for genetic material. And this will be something that we'll see often, and as we go in the next few minutes. Compared to that, you have the other cycle, that is the one that takes place in mammals and birds, where there is carnivorism, and they also consume the feces of the infected cats. And here is totally asexual. It's just clonal. It's just one strain reproducing itself over and over time. So this has led for years since the discovery of Toxoplasma gondii as a species in 1908, that it was a single lineage, that it was just one parasite, very homogeneous clonal expansion of the organism. And it was in 1995 when Hoey and John Boothroyd's group, they found that there were primarily three lineages, types 1, 2, 3, in this 1995 paper, where they thought that it was just a simple three, let's say, variants of the parasite, three lineages of the parasite. And at that time, they used restriction fragment-length polymorphism, RFLP, as the main technology to find out that this was the case. It was later found that it was not the case, that in this case, they had used an overrepresentation of samples from Europe and from North America, and that the technique that they have used, the RFLP, it was important at that moment. This was a revolution in terms of the genetics of the parasite, but it was not with a high resolution enough to distinguish other variants within these three lineages. So the next thing that comes is that we have a report in 2011 that there was a fourth clonal lineage of the so-called type 12 variant that was found primarily in wildlife in the United States. And that was found by Kahn in this paper. And what it became obvious is that there was a geographical distinct separation in the genotypes of the parasite, where in North America and Europe, there was an overrepresentation of the type 2 strain. So it's a theme that you start to see, at least in this paper so far, that the type 2 was having a predominant role in North America and Europe. There is something that happens in North America later that I'll point to you in a minute. They also saw participation at the strain type 3 and the type 1. The type 1 is the classic strain that is very aggressive, very virulent in mice. The main representation there is the so-called RH strain. So if you think about an aggressive strain, always carry in mind as a reference the RH strain. This is the strain that kills mice within 2 days. The type 2 is less virulent, and is the reference strain is the ME49. The ME49 is a strain that you give to mice, and mice live for the age of the mice in general. So that's the classic type 2. And type 3 is considered of low virulence. So it's found here that in North America and Europe, there are these strains that tend to stay for long periods, lower virulence, and they have to have a clonal expansion. It's just one lineage, one variant of concern doing the genetics. And in contrast, in South America, this is a great genetic diversity. So the more separated these lines are here in the left lower, the more distant are these strains from each other. And here there are many strains that don't follow lineages, that they don't follow clonality, and they tend to be called atypical. And in animal models, they tend to be more aggressive. More in this in a few minutes. And what it was really found and continues to be found, despite that we are having a greater understanding of the greater diversity, is that the type 2 is really the predominant strain in France and Europe. Then something happens in 2012, just a year later, where Su and his group and Duby and others find that there are actually 15 haplogroups defined in 6 groups. So 15 haplogroups is like a subgroup, and 6 clade is like a genetic group. And in this case, they use almost 1,000 strains. And out of the 1,000 strains, they found that there were 138 that were representative. And they were very clever. They used several different genetic methods. They used the restriction fragment, length polymorphism, the RFLP, but they also use microsatellite markers to complement the findings of the RFLP, and they use sequencing to find that exactly where each strain fell. And they found that actually there were 15 genetic types, so-called haplogroups, and you could group them in 6 clades. So you can see here how the type 1 strain that is in the haplogroup 1. Luckily, the type 1 original that we had falls into the haplogroup 1. In this case, they use a different aggressive strain called GT1 that's similar to RH. And you can see here how in the haplogroup 2 is the famous ME49 that is the type 2 originally. Luckily, type 1 is in the haplogroup 1, type 2, haplogroup 2, type 3, haplogroup 3. And the ones highlighted here in yellow are the representative strains within each haplogroup, within each clade. So in summary, what has been found since 1995 is that the parasite has a genetic that is way more diverse than it was thought originally, but still Europe carried that signature where the parasite is highly homogeneous, clonal, whereas in contrast, in complete contrast, in South America, it carries that signature of high genetic diversity. Second here, great. This is a study that came from Denmark, and its first author is Dr. Jokelainen here in the group. And what they found is they were able to type the strains from clinical samples in humans, and they found that 72% of the strains, as expected, were of the type either 2 or 3, that it has this lower virulence. In this phylogenetic tree, you can see first that there are 16 haplogroups from 15 from the previous paper, so 16 haplogroups. And you can see the dark black triangles, the strains from their clinical samples. And you can see that most of the cases the black triangles fall in the haplogroup 2. Those are patients who have either central nervous system disease or ocular disease. You can see how here there are some patients in the type 3 that it also tends to be of lower virulence. And you can see, however, that there were some patients in the black triangles that fell into what is called the Africa 1 strain in the haplogroup 6. So they were able to confirm that there is a type 2 predominance. However, that one has to be careful because it highlights that clinical toxoplasmosis can also be caused by genotypes that are usually not seen regularly in endemic regions from patients having traveled from imported food, as it has been demonstrated in numerous times. So in general, your patients will follow the genetics of the parasite in your area, in your region, but you may have to also be open to the possibility that they're infected from with strains that are not from your area. This is a phylogenetic tree of strains infecting patients in the United States that we published 3 years ago, where we found there were a type 2 representation similar to Europe, but there is a significant representation of atypical strains, like the ones that are seen in Latin America here in yellow. So in the US, primarily we see about 44% type 2, 44% atypical, and about 12% of the type 3 that is of the lower virulence. So the US appears to be in the middle between like some of what is seen in Europe, some of what is seen in Latin America, and 10% of the typical strains belong to the so-called haplogroup 12, that as soon as you hear haplogroup 12, your mind should race into wildlife, organisms that are coming from deer in the wildlife. There was a report by John Boothroyd and Michael Grigg that the strains that they were able to get from patients having ocular disease, not just focus on the ocular disease, that they were more atypical than expected. And this caused a lot of uproar for the possibility that the parasite had some tropism for the retina in terms of the atypical strains affecting more the eye. There was also this report from Brazil about the strains affecting the eye and how they were so genetically diverse as expected from Brazil. So infection with toxo in Brazil is more likely to lead to more severe disease and more recurrences, probably connected to the fact that the diversity is greater there. There was then this report from Germany where they found that the patients with ocular disease, at least the majority of them, were infected with the type 2, which is expected. In Europe, it is lower virulent strain. So you start to get the picture. Are the strains preferring the eye genetically? Some strains genetically preferring the eye, or is it just that the eye is representing what is also present in the population? This is work from Latin America, from Drs. Alejandra de la Torre and Dr. Jorge Gomez Marin, and by our dear Hermano Candolfi, who also passed away last year, and we miss him terribly. In this work that was done by them, they found that of patients with ocular toxoplasmosis, they were able to actually sample the aqueous humor that it had been sampled for clinical purposes, and they measured cytokines there. And it was fascinating because these are patients with cataracts from France, cataracts from Colombia, so those are controls. And these are patients with ocular toxoplasmosis in France and ocular toxoplasmosis in Colombia. And they found that interferon levels in the aqueous humor of ocular toxoplasmosis patients were lower in Colombia than in France. And also that the levels of interleukin-17 were lower in those patients from Colombia as well. And these are two primary pro-inflammatory Th1-like cytokines that are key for defense against T-cell mediated pathogens like toxo. And they found, interestingly, that the TNF was in the opposite. TNF in the patients from Colombia with ocular toxo, the aqueous humor levels were higher, suggesting that this probably could be mediating the immunopathology that Dr. Garwell was referring to you in his presentation. And they confirmed that primarily if they were able to genotype, they were genotype 1, the aggressive type, atypical, the aggressive type. And in the serotyping, the 2 was predominant in Colombia, and the atypical was atypical in Colombia and type 2 in France. In summary, toxo in South America seems to have a for sure, and Dr. Belfort will confirm this presentation that is more aggressive. Is it because they have more reinfection? Is it because there is more virulence, like the rupturing protein 18 that has been associated with greater protein? Is it because more atypical strains are present? And that can lead to more severe presentations and more relapses? Is it because there is more natural drug resistant, like Dr. Jorge Gomez Marin had reported in the dehydroteroase enzyme or in the dehydrofolate reductase enzyme? What about Asia? Asia also has been represented, though more modestly, in studies. And it's fascinating because if you see Asia on the west coast, in the western part of Asia that is closer to Europe, you see more representation of the type 2 strain, which is you expect because of the geographical proximity with Europe. So that green is the type 2 strain that you tend to see more in the western part of Asia, whereas in the eastern part of Asia, in red, you see more the type 1 strain here that is closest, separated by the Pacific Ocean in terms of proximity to North America and to Latin America. And lastly, what about Africa? Africa is the continent that unfortunately has contributed with the least to the genetic studies of toxo, though talking about genetics and organism, it is South Africa now who's leading the effort of genetic typing for the Omicron, for the SARS-CoV-2 pathogen. So we hope that this will lead Africa to also do that, the same thing for Toxoplasma. And you can see in Africa how in the west coast of Africa, you see this Africa type 1 variant that is seen actually in Denmark, as we saw recently from Dr. Jokelainen's study, or you can see that also represented in some studies in Brazil. And you can see how in the east coast of Africa, you can see how the type 2 strain in green here is more represented because of its proximity to Europe. So in summary, it's an evolving story. It's a fascinating story. There is no question that there is a lot of genetic strains there, but that in Europe, it continues to be the fact that there is a predominance of the lower virulent strain, the type 2. There is no question that in South America, we have this wide genetic diversity and the presence of atypical strains. In North America, we have a mixed picture. We have about 44% of the type 2 seen in Europe, and type 3 and atypical of the type 44% atypical of the type seen in South America, so it's half and half. And in Asia and Africa, there are different population genetic structures depending on the geographical locale where you see in those continents that are actually vast in size. In summary, it is unclear whether some strains exhibit tropism for the retina. Are strains preferring to go to the retina versus other organs? But it is sure that the greater diversity has been associated with more severity and more relapses, and that appears to be associated with the atypical strains. Thank you.
So Jose, thanks so much for this really fascinating topic. Amazing. Thanks a lot. Next one will be Rubens Belfort from Sao Paulo, talking finally about the therapy for ocular toxoplasmosis, systemic therapy, intraocular therapy, and last not least, prophylaxis.
Thank you very much. Yeah, thank you very much. Can you hear me?
Perfect.
Oh, great. Oh, so thank you very much. Good morning, good afternoon, good evening. It's a great pleasure to be here today and to present you some of my experience. Next. I just realized when I was preparing this talk, next please, that I've been working in the field for half a century. And in all these years, I believe that I learned to be very humble regarding toxoplasmosis and also specifically to the treatment of ocular toxoplasmosis. Next one. I want also to take this opportunity to thank the IOSG to keep the high level of science and education in UVEITIS, and specifically to thank Manfred for keeping IOSG alive and productive in all these pandemic times. I believe that Manfred Zihurt has been probably the best president that IOSG has had in all these 40 years that I followed this organization. And thanks to him, we have this now international symposium on toxoplasmosis that I find very useful because next, toxoplasmosis for most of the countries is one of the most important UVEITIS entities. Next one, please. And of course, it's been known for a long time. In century 19, it was considered just a genetic change, and we learned a lot, but we still rely on very old information regarding the practical aspects of how to treat this infection. Next. And this has been my favorite slide in the last 30 years. We know little, and much less is true. Next. When we go back to see what happened in all these years, 1950s were very important because medicine advanced a lot. Steroids, pyrimethamine, sulfadiazine were starting to be used, especially steroids. And in the 60s, all the UVIs were basically treated with huge amounts of steroids, and including toxoplasmosis. It was in the 70s that better understanding of toxoplasmosis started, with also the results of serology and the steroids concern. And we start to use the same drugs more and more that we use nowadays. Next one. We are not very powerful regarding the treatment of ocular toxoplasmosis. Take this into consideration. Next. And also do not forget that very little that we say and we do is based on deduction. We still rely a lot on induction and seduction. The industry that helps a lot also tries to sometimes impose their needs on us. It is very important to take in consideration all that when we learn and we decide on what to teach and how to treat the patients. Next one. We're gonna hear more about vaccines, but as far as you know, there is no vaccine in the horizon for toxoplasmosis, and it will continue to be a major problem. Also, with globalization and migration and huge number of people going from one country to the other, one region of the world to the other, we're gonna get more and more a mixed situation in which a specific local region cannot represent what goes on the others. Next. Next. Very often, we see more and more with advance of technology, lesions that in the past were not recognized. And we've been learning a lot on that, not only because we need better diagnostic tools, but also they learn and we learn from them and teach us how to treat. These are patients in which in the past were probably not well diagnosed and right now are easily seen, and we can act on this visual information in order to plan our treatment strategy. Next. When we talk about treatment of toxoplasmosis, of course, it depends on the situation itself. I'm going to show you now some examples in which probably we will treat it very different from one case to the other. Next. This is one example that, for instance, I would say that we will need anti-toxoplasmic drugs and systemic steroids, and even without that, not easy to have a good result. Next. Other example, patient that had herpes and later on was also diagnosed as a positive IgM for toxoplasmosis. And there are some cases in which the diagnosis is not very specific, and we end up treating more than one infectious disease in order to try to do the best for the patient. Next. Also, a patient with positive IgM, no clinical symptoms in the eye, no clinical symptoms outside the eye, and you identify a very small lesion that does not need to be treated. But is it a place where future recurrence will happen? Should we treat patients like this in order to try to avoid recurrences? Next. Well, we know that the clinical diagnosis sometimes is easy, but not always. And according to the clinical diagnosis, we go from one treatment to the other. Next. You see here patients that had bilateral acquired toxoplasmosis and were treated, and 6-month follow-up, they're completely normal again, except for the scar. Next. Other patients, though, have a very different clinical picture. You see this one here, where the right eye would not be very specific of toxoplasmosis, but in the other eye, there was a very typical scar. And this patient later on developed very severe vitis reactions, and the vitis became completely cloudy, and it's not even possible to see the fundus of the eye. So when you also plan the strategy of treatment, you have to get information about what happened before on that patient and how the patient is evolving in order to give the best future for him. Next one. Well, it's already been talked about the difference of toxoplasmosis in several parts of the world. And this is what we see very often in toxoplasmosis in the Amazon. We see completely different cases in the Amazon when we compare to the southern part of Brazil. Next one. Also, we talk about toxo. We should not forget that some patients, not very frequent, become patients with anterior uveitis, and Fuchs heterochrome cyclides has also been identified in ocular toxoplasmosis in patients that have no antibody for rubella and negative for the other differential diagnosis. Next. Well, there's some false dogmas in toxoplasmosis that I think are very important to understand. In the last decades, we have learned that it's not true that our cases are congenital. It's not true that they must present retinochoroiditis. We have learned that pregnant can transmit the disease sometimes for more than one fetus. We have learned that cats and meat are not the only source of the disease. And very specifically, cats and meats and also anti-toxoplasma treatment should not be given just for 4 to 6 weeks. Depends on the case. I remember in 1974, I once asked Richard O'Connor why he was teaching that we should treat patients with toxoplasmosis for 4 to 6 weeks. And Dick O'Connor said, well, people start to ask me, and I had to have something to say. So I said 4 to 6 weeks, but I personally do not believe on that. Well, this was Dick O'Connor in the 70s, and people started to repeat it and repeated it without having very solid information about that. The video that you saw here on the right was a video that Herb Kaufman gave to me, and a video that he made in 1960 showing a toxocyst rupture and the huge amount of organisms that can come after it happens. So we also have learned that we need to develop drugs that prevent and eradicate ocular disease. And something that is not still being realized, but personally I believe is very important, is that ocular recurrences are not only related to the rupture of the retina cyst. It's very probable that most of the symptoms and signs that you see in ocular recurrences in toxoplasmosis depend of extraocular mechanisms and extraocular cysts that rupture in other parts of the body. Next one. And all that has to be considered when we talk about recurrences. Next one. So when we talk about treatment, take in consideration many things, active or inactive. We're talking about the cure of the active disease or prevention. We have to talk specific about what to do in pregnancy, what to do if patients are allergic to drugs, and so on. So the treatment depends on the age, on the size and location, the inflammation severity, and also the vision involvement. We should not try to treat toxoplasmosis without paying attention and all these aspects. Next one. Next. Okay. Next one. Next. Okay. Next one. Going to be better. Okay. When we talk about toxoplasmosis, first of all, I have to talk about anti-toxoplasmic drugs. and then steroids. And when we talk about steroids, local, systemic, and periocular and intraocular. In the 70s and 80s, people were using a lot of periocular steroids. And then came the information that periocular steroids could make the infection worse. And periocular steroid was banished from the treatment of ocular toxoplasmosis. With the development of intraocular medications, I believe that there is now a tendency to understand better and maybe to use more intraocular drugs than in the past. Next one. I read a comment about not to treat all the patients from 4 to 6 weeks. And it always depends on the clinical picture. We know that in many patients, the treatment of ocular toxoplasmosis with anti-toxo drugs should be done for many, many months in order to try to keep the patient without recurrences and decrease the chronicity of some of the lesions. Next one. Reinfection, I believe, is a possibility. And we have now patients that we've been following, patients that move from the southern part of Brazil to other places, and then start to have much less recurrence than they were having in their original places. And so there is a possibility that in some hero areas, reinfection may play a role and may explain why the pathogenicity is higher there in the other places. Next. And then we go to the specific treatment, and pyrimethamine and sulfadiazine is 1955. And we're not sure that the drugs we have now are better than the old ones, and we're not sure either that they are worse. We have learned in the last many years that there's no advantage of using quadruple therapy, and also the AIDS experience has told us that usually we do not need a cocktail of many drugs to control toxoplasmosis. Next. Okay, I'm just gonna show it quickly because it's gonna be taped. Pyrimethamine, should we use the concept loading or not loading? Loading was information that appeared with Herb Kaufman when we worked with monkeys. And in monkeys, he showed that there is an advantage of using a loading dose to treat the toxoplasmosis. Clinically, I don't think that is considered very important to load the patients like that, but maybe in some specific situation, we have some advantage. Next. And this is a paper, 1959, when he published it. Next one. Also, folinic acid is something that is being used and is being repeated in every textbook, but the truth is that it's very rare. And in most of the cases, a good clinical following of the patient can tell you the ones that have a higher risk to develop it or not. Next one. See, here's one of these rare examples with complication of pyrimethamine treatment for ocular toxoplasmosis. Next. Sulfadiazine, of course, is important, but compliance is very low. It's not easy to take so many pills for such a long time. And also allergic reactions may be important in the real life. Next one. So we decided many years ago to use more and more Bactrim. And with the good clinical impression and the good clinical experience that we had, allowed us to go ahead on that. And in our department, later on in many parts of Brazil, Bactrim became our first line of treatment in ocular toxoplasmosis. Next one. Clindamycin was something that in the past was very well considered, and there was an impression that it could eradicate the toxoplasmosis, could kill diseases, but we know that this is not true, and also has some complications, and is not considered the first line for the treatment of ocular toxoplasmosis. The advantage of it is that it can be used also in intraocular injections, as we'll discuss later. Next. And then comes, of course, steroids, and 40 to 80 milligrams a day. I believe that everybody agrees should never be used without anti-toxoplasma covering, and tapering is very important. More and more, we've been considering the possibility also of using in some patients intraocular injections. Next. Always take in consideration that ocular toxoplasmosis is part of a systemic disease. This is a pregnant woman with congenital toxoplasmosis. The baby was born with a very severe disease, and she also had the ocular infection in both eyes. So in case like that, of course, the treatment is different than the ones that we have, and goes back to the point I tried to make. The treatment of ocular toxoplasma depends on the eye and on the patient. Next one. Of course, applies this also to the patient that have AIDS, in which the disease presents a very different way. Next one. We all that, next one, please. I apologize for that, but we had technical problems, and I asked the people to help me on that. So going back to AIDS, we know that heart and ways to improve the immune system helps a lot. And many patients with ocular toxoplasmosis do much better when they have AIDS and they are controlling the HIV infection. Next one. You see here some examples also in patients that have scar, CMV, very low levels of CD4, and never develop an active lesion of toxoplasmosis. Why does it happen? Well, maybe in this situation, there were no more toxocysts to rupture, and in spite of the low level immunity, no activity of toxoplasmosis, but very often is the opposite. Patients develop ocular toxoplasmosis, and very common also to have an infection in the brain. Next one. Well, photocoagulation. It was used in the past. Next. May have some indication, but not usually good enough to prevent recurrences or to control the disease. Next one. Also because you never know where the recurrence will happen. And also patients can be inactive 10 years and later and after have 1 or 2 recurrences. Why does it happen? We don't know. Next one. But not forget that all important to follow these patients. And yes, photocoagulation should be used in order to avoid the retinal detachment that very often makes the vision much worse in these patients. Next. Now to conclude my talk, I'd like to discuss a little bit about how to prevent toxoplasmosis. This is a medical student, now a physician, that had many recurrences and start to have take Bactrim 3 times a week, and decided, now he's a physician, decided never to stop it. And he's been using Bactrim now for over 12 years, 3 tablets a week. Next one. And this is the paper that we published in 2002, trying to establish if Bactrim could have help in avoiding recurrences. Next one. One tablet of Bactrim 3 times a week for 20 months. Next. Showed the protection 70%. Next. Next. And the conclusion is that we should use it. Next. And it was reviewed by many people. Other papers also confirmed that. And we use it, not, of course, in all patients, but the patients that are at a higher risk. And very important in children. We believe that one of the worst situations is that when you diagnose oculotoxibus in a child and inactive, sometimes one eye is lost, but the other has good vision. And then the child comes back after 1 or 2 years with severe disease, both eyes. So especially in children, it's very important to try to develop something that would decrease the number and severity of recurrences. Next. Well, we went back to the same patients and studied what happened after they had stopped the prophylactic. And we found that, unfortunately, the long-term loss of effect years after the drug was suspended. So it's not easy to say, but we do not know for how long the patient should take this medication in order to have less recurrences. 1 year, 2 years, forever. Again, would depend on the situation and the eye. Next. 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 We know that intraocular drugs can be used, and over 10 years ago, we showed that clindamycin and dexamethasone could help in some patients. Next. And these are some examples. Patients that have a disease, 1 injection, and the disease went away. Next, we have now a large number of patients that received that, and we believe that has a poll also for that. Next. We decided to work with other anti-toxoplasmic drugs, and we started to use also Bactrim injections inside the eye. Usually we did it just Bactrim, and with the good experience that we had in patients that were also recovering using systemic anti-toxo drugs, we started to use the association of Bactrim with dexamethasone as an alternative therapy for ocular toxoplasmosis. You see here a good example of that. Next. Before injection, 2 weeks after injection, and 4 weeks. This patient was treated only with 1 injection of Bactrim associated to steroids. Next one. And then we did something that was very wrong, and I bring this to your attention because we learned a lot with that. Next one. We decided that we should add triamcinolone to Bactrim in patients with ocular toxoplasmosis. And in 5 patients, initial patients, they all experienced a worsening of visual acuity and intraocular inflammation. So it's very interesting and very sad. We learned that triamcinolone, maybe because the long-term effect would make the uveitis much worse in this patient, even with the coverage of anti-toxo drugs, showing that probably there is a very important infectious role in these patients. Next one. And this is my last slide, the sad story. Of course, we stopped the trial, these patients were treated, but we lost vision in 1 eye. Next one. You see here 3 weeks, 4 weeks after the injection, in spite of full anti-toxo systemic treatment. Next one. And we are now using, doing other studies in toxoplasmosis. We can maybe talk a little bit about that in the discussion. We'll talk about the different effectiveness of treatments of ocular toxoplasmosis in real life. And also we're trying to develop other therapeutic strategies for patients that have recurrent toxoplasmosis and are allergic to sulfadiazines. What could be used in order to try to decrease the recurrences? Well, thank you very much for the opportunity of being here. And I thank all the speakers for the excellence of this meeting. Also, thanks to the global people that helped me with this presentation. Thank you.
Rubens, we have to thank you. Wonderful presentation, very conclusive, very, very helpful. We're running now a little bit out of time, but we are reaching the last one, the last of the schedule. Jorge Gomez Marin talks about a new perspective. Is it possible to create a vaccine for Toxoplasma gondii? Jorge.
Yes, thank you very much, Manfred. Thank you for the organizer for the invitation. I will try to go very fast because we don't have we are very early in this morning. These fantastic talks have been a very good background for, I will try to give some key message about what is about the possibility of a vaccine for toxoplasmosis. The first meeting of the World Health Organization was in '92. But you can see here some big names in Toxoplasma, like Jack Remington, but also David Sibley was there, and Louis Weiss, for instance. And the next year, we see here appear the first commercial vaccine. So it's a very fast talk because, yes, it is possible to have a vaccine for toxoplasmosis, but the problem is the vaccine is for cheap. It's the only vaccine you had available to use for toxoplasmosis. For us that we treat patients, human patients, we know the urgency to get a vaccine for human toxoplasmosis. So for me, the question, the challenge is, it is possible to have a vaccine to be approved for human toxoplasmosis, and how far for a trial humans. So I will try to explain the problems to get a vaccine for human toxoplasmosis. And for this, we can see here which anti-parasite vaccines are currently available. And you know, this year was approved the first vaccine for a human parasite, the most important parasite, because of the number of people affected, the mortality that induced is a real problem for humanity, the malaria. But when you see what indeed was approved, was a low efficacy vaccine. Only 30%, 36% of efficacy. Maybe for some of you can be astonished how it's possible to approve a vaccine with a very low rate of efficacy. But when you see the information that the World Health Organization gave about why it was important to approve a vaccine, it's a commercial vaccine developed by Glaxo, RTS, you see that indeed it's only one part of different measures. So that is getting along the reduction in mortality is indeed a part of one with different mechanisms. For me, so it's important also to do justice for history, because maybe do you have listened about the vaccine developed by the Colombian group led by Dr. Patarroye, that is a pioneer in the peptide-based vaccines. And his vaccine achieved a low efficacy of only 30%, and he donates this vaccine to the World Health Organization. So maybe you can see that this was proposed in 1996 by the same Alonso that leads the program of the malaria vaccine currently approved. And he, in that time, 1996, also suggests that a vaccine with a low efficacy as 30% can be cost effective. So you can see that it is possible that even a vaccine with a low efficacy can be useful in terms of public health objectives. So why I am speaking also about malaria vaccine? Because that we can tell us a vaccine will be successful in human is the natural history, how we our immunity acquired naturally can be protective or not. But in the case of malaria, you see that in malaria exists a phenomenon that is called the premonition. Premonition is a partial immunity because you can get new infections and you can be symptomatic again. So it's a partial immunity because the severe cases are reduced, but you're still infective for malaria, for Plasmodium. In the case of Toxoplasmosis, here I'm showing you this very beautiful diagram picture done by Dr. Frankel, who discovered the life cycle of Toxoplasma, and he was a pathologist. And you can see here the correlates what is happening in the human in the case of the immunity in a normal immune host that achieve the protection, and then the tachyzoids become a bradyszoids. So we are not able to eradicate the parasite from our tissues, but we get immunity against new infection, against new symptoms. And even you can listen, Dr. Belfort, because in Brazil it has been reported the reinfection. Indeed, this is a very rare event. So for the vast majority, and in terms of public health, we can get immunity after a primary infection. That is very important concept because that tells us that it's possible to achieve a protective immunity that can be useful in terms of public health goals. So here I will try to tell which are the strategies we have now to develop a vaccine for Toxoplasma that has been already tested in animal models. So the first one that also was tested by Dr. Frankel, the TS4, was a strain attenuated he used in cats. But the problem with that kind of strategy is the same that occurs with the polio virus. The vaccine for polio can revert to a phenotype, so induce a paralyzed vaccine paralysis induced because the vaccine can revert to a pathogenic phenotype in some case. But we know, but in the case of polio, this in terms of cost efficacy and efficacy for the public health program goals, is useful to use that kind of strategy of development of vaccines. But in the case of Toxoplasma, also there are the problem of the cause of the culture of parasites. So this is also the problem to total antigen extract preparation, because you need to prepare and to have a lot of parasites culture. Also, there are the problem when you use the extract from lysed parasites, is the standardization of the method to prepare that antigen total lysate. So the next possibility, which is very attractive in terms of how to develop a protective vaccine by using only selected proteins of the parasite. So, the problem then, I will try to show you in the next slide which are the main problems in the case of the parasites. We can obtain a very good immune-specific response with kind of vaccine candidates. Now we have the COVID vaccine, and they are proof that it's possible to obtain vaccines based on nucleic acid platform. So in the next year, probably we will see very good vaccine candidate for toxoplasmosis based on nucleic acid acid platform, even with RNA or DNA. Another possibility is to use bacterial vectors. You include plasmid coding for some proteins of the parasite, but here again the problem is in immunocompromised hosts, because that kind of vector can be pathogenic for immunocompromised people. Is the same with viral vectors. So that is one of the limitations with that kind of strategies. But that is important to know is that for a useful vaccine for human toxoplasmosis, we need that vaccine induce a specific cytotoxic CD8 T cells, and also as a very important response of CD4 memory cells, because we need for the memory for the long-term protection. So this can be achieved, and it can be shown that with any of the good candidates in the mouse model, we can achieve that kind of immune response in the mouse. And so the interesting with the subunit vaccine is to choose the proteins that can be protective. So this is a very big challenge, because protozoa, that is one of the problems with the parasite, had a lot of antigens, most virus. So that means that it's very difficult to identify which antigen can be used. One of the problems is that the system for invasion, for instance, have a redundant system. That means that the parasite use, for instance, in the case of micronems, a different proteins that have the same function. The same occurs with the surface-related sequence. So the SAT1 or SAT2 that has one of the first proteins were used in the models or in the animal models to test vaccine. So here, this is an elegant experiment by Dr. Dubremet, now retired. He is a parasitologist in France, very important in his discoveries about the proteins of Toxoplasma. And he used knockout strains. And you can see here, when he used only a gene that was invalidated, the survival of mouse was not important. But when he made double knockout for the 2 micronem proteins, in that case, he obtained a very good survival of the mouse. And the same, of course, with the SRS proteins, where we found the SAT1 and SAT2, and here again, the same type of experiment. This is a paper from Dr. Grid, who works a lot in this kind of proteins. And you can see here, but to improve the survival of the mouse, you need to use a double knockout parasites. So it showed that the parasite had a very sophisticated system to invade it. That explains also the success of Toxoplasma, because have a very redundant system to invade the cells. So now we had a lot of very good candidates to be used in subunit vaccines. And also we had improved the production of the protein recombinants or the nucleic acid platform. So here we had a very good amount of proteins that can be used as vaccine candidates. So one of the things that we can use is to dissect within the proteins to identify which region of the proteins are important in the protection. And we made this in our one of our first boards working for the vaccine candidates with the group of Dr. Pat Arroyo by using the P30 protein, the peptides were derived from this protein, and the first amino acids from the protein were protected. In the contrary, the last peptide, the last amino acids, obtain a very good protection in the mouse model. But the importance of peptides is because they are cheaper to produce. But the problem is that each peptide can be recognized differently according to the haplotypes. So that explains why the vaccine based on peptides had problems to be protective in the most of the people. You need to identify peptides for each haplotype. So you see, it's a very big challenge with the vaccine based on peptides. Dr. Maleo in Chicago developed a strategy called immunosense, a rational approach, by selecting peptides that were identified for a specific haplotypes. So we know now there are 5 super haplotypes. If you construct if you make a construct of peptides for each one of those haplotypes, you can reach the 90% of human population, according we know about the distribution of those haplotypes. So the problem is to construct a chimeric with a lot of peptides with different proteins that should be protective. So for this reason, we work with my student at that time, Dr. now Nestor Cardona, that work in the Universidad Antonio Nariño, a pipeline in order to select which sequence specifically can be immunogenic, but also recognized for a specific haplotypes. And by using this strategy, he could found one peptide that was recognized for the people with A2 haplotype, and he showed that it was inducing an interferon gamma response. So this shows us that it's possible to found peptide by using the bioinformatics that can enable us to study very fine, to do a dissection of the immune response very fine. In this way, we can achieve a vaccine that we can identify which people can give an immune protective response. So this is you can see the difficulties is you can want to use a strategy based on peptides. But also it's important to know that we don't have in humans the same effector mechanism of the mouse. So for instance, we don't use the nitric oxide as mechanism, effector mechanism against Toxoplasma. So for that reason, we developed a model with human cells. Recently, it was confirmed that we know now that we use different pathways in the effector response by interferon gamma in human and in mouse. So the effector mechanisms are very different. For that reason, it's not possible to correlate all the results or to extrapolate all the results in mouse for human. And for that reason is that we developed an ex vivo model by using human mononuclear cells. And with that model, we could identify, Alejandra de la Torre showed this previously in her talk, and I will show you here, with this model, we can examine very fine a dissection how the peptides induce a specific cytokine response. We we know that we need a Th1 immune response to be protective. And we can see here that for the children infected congenitally, we could change with a specific peptides the immune response from Th2 cytokine response to a Th1 with a specific peptides. But in the case of the ocular toxoplasmosis patient, it was not possible. So we need to go deeper, and the problems we know now that we have many genetic polymorphisms associated to the susceptibility to ocular toxoplasmosis, to the recurrence, and we are now working to know how to change that shifted deviated response, immunoresponse. So that is promising for the immunomodulation in the future. But this is the last slide. I am showing you here our last strategy by using our ex vivo model in order to identify, in that case, the company Sinano in Lille is working with us in order to show if with a total antigen preparation with nanoparticles can induce a specific immune response, and with the induction of the CD4 and CD8 specific immune response. And in this case, we show that it's possible to induce that subset of lymphocytes in people with different serotypes. So that is very important because of the presence of atypical strains in our continent. So also it's important to think that we maybe not need our only vaccine, because we will need many vaccines. Once to prevent congenital infection, in seronegative women, we can use live attenuated, but during pregnancy, it should be preferred recombinant protein fraction, nucleic acid vaccines. The same in immunosuppressed people, but also one of the major goals we have is to find how to induce in prevention of the reactivation in patients with ocular toxoplasmosis. So in conclusion, I think it is possible to have a vaccine for human toxoplasmosis, but we have now many promising candidates that can be essayed, but I think indeed the real requirement we need now that is lacking, because we have now very good candidates, is the funding. So thank you very much for your attention. And I only want to thank all my team in Colombia that made all this possible, particularly Alejandro Zamora, who worked Laura Garcia, who worked in the last experiments with the nanoparticle vaccine candidate, and all the team working in the immunological assays, of course, working with Dr. Alejandra de la Torre in Universidad del Rosario. Thank you.
Jorge, thanks so much for this highly informative talk. Fantastic. Thanks to all of you very much for this talk. We are running nearly out of time, which is sad because we still have actually some kind of controversies. The idea of a controversy is that we try to ask a few pointed questions to the experts about things which are not really 100% covered by the literature. Let me limit this to 3, I think, more or less clinical questions. And if you have to talk about this one, if you would love to talk about this one, then please very, very short answers. Number 1 is, what is the role of AC tap in contrast to vitreal taps in detecting antibodies in ocular toxoplasmosis? Do you prefer that?
Maybe give a short response to that. is Vitrus is probably more sensitive with regard to PCR, but much more difficult to access because you do Vitrus tap only in doubtful cases or in retinal detachment. So the standard is aqueous humor tap, and antibody testing in aqueous humor is as sensitive, at least as sensitive as that of Vitrus tap.
Rubens, would you agree, more or less?
I agree.
Perfect. Second
That
question.
was a good short answer.
You're muted, Rubens.
That's it. I agree.
Okay, next question. Toxoprophylaxis. Rubens, you gave a wonderful presentation already about reactivation. What about surgery? I think there was some meeting in Tel Aviv where most of the people suggested to use anti-toxo treatment during surgery, even cataract surgery. What do you think about that one?
It's a very controversial aspect. In Brazil, 80% of the patients have scars in the retina, and we almost see no recurrence of ocular toxoplasmosis. We never use anti-toxo medications when a patient is going to be operated of cataract, unless there is a chronic uveitis.
Yes,
I agree. That's also my experience.
Perfect. The final question is about pregnancy and toxo infection, congenital toxoplasmosis. So you see a pregnant woman infected with toxo. When do you and how do you check these kids?
You can repeat the woman?
Where's a pregnant infected woman? How do you check these kids if they have toxoplasmosis, ocular toxoplasmosis? Do you check them regularly, very, very early, or you take your time, or you wait until you see some first clinical signs?
I think it's necessary to do many examinations. Huh? It's not only the early, but it's necessary also a follow-up. It's very important.
When do you start?
I think within 4 months, 4 weeks after birth, one should have a fundus imaging done because it will have consequences for treatment. If the child is without symptoms and manifestations, it would not necessarily be treated for 1 year. But if there is ocular affection and cerebral calcification, then definitely a treatment is recommended. And this is what you need for guiding. You need to look for calcification and you need to look for ocular affection to guide treatment.
Are there guidelines available for such situations?
There are some, in fact. If you look inside the pediatric literature, there is guidelines. One other thing might be to test the child or the placenta when the child is born, and lesions can take up to a year to develop. So prophylaxis is recommended if there's been any kind of seroconversion in those children for about a year.
I think that's a problem. They can develop up to 10, 20 years after this one.
And it's important to follow them all the way until routinely they can verbalize the lesions, because the clinical manifestations, they can be silently present in those children.
Rubens?
If I can add something about what Dr. Montoya just spoke. Yes, non-verbal kids, very important to follow them. And more than that, these are the kids that I really tend to use prophylactic treatment forever. I get so frustrated, desperate when I see a child that lost the vision, the second eye, because the recurrence was missing. The parents feel guilty. So we have to really approach them very close.
Pika?
I would add to this that exactly this is one of the groups where we should not say this is asymptomatic Toxoplasma gondii infection, because there's limitations what we can find clinically, and then rather think that way. So I would echo these comments very strongly.
Wonderful. So probably we'll be able to establish such a working group as mentioned to all of you. I will try to invite you with the goals and these things in the next few days. Well, we are coming to our end. I think we have 10 minutes over time. Would love to thank you so much for this super wonderful meeting. Rubens, a comment?
I'm sure that I speak on behalf of everybody. Thank you so much, Manfred and Mark, for keeping the ISG alive and having this meeting. Thank you very much. Happy Christmas to everybody.
Happy Christmas to everybody.
Thank
And I
you.
think such a meeting
Jose
shows
Montoya,
exactly.
Jose Montoya, I don't know if you slept or you just had a typical Latin American evening.
No, no, it was announced 5 a.m. my time.
Perfect.
It was
So we
announced
tried-
5 a.m.
We try to
Yes.
be very, very accurate with these timings, but you know, they change every half a year and there must be something went very wrong. I'm super, super sorry about Jose about these things. Yes. I would love to wish everyone a wonderful Saturday and Sunday. I hope you enjoyed that. Again, if you want, you will receive the evaluation sheet, send it back immediately. In a week, you will get all these documents which you need for CMA points. We will meet again, I think it's the 12th, something like 12th of January. And then we'll have a very wild webinar about how to start a uveitis clinic. And that was an idea of one of these participants, probably I think from Asia, and I think it's a very, very good idea, and we want to bring up a very basic information about this one to help you to become even better. All the best to you.
Thank you so much,
Bye.
Manfred. Great webinar. And thank you to all.
I love it too.
Bye, Mark. Keep kicking,
Bye-bye.
Mark. Keep kicking.
Thank you.
Ciao.
Thank you.
Bye.
There are still few questions to answer. I see that. Rubens, can you take the first one? In the absence of pyrimethamine? Dipankar
In the
does.
absence of
Hi,
pyrimethamine,
Dipankar.
what is the idea association to teacher? I go to Bactrim.
Can you add this? In
Sure.
the things? Wonderful. So the other ones are all answered. Enjoy the pre-Christmas
you.
time. It
Please
was lovely
send
to
the
meet
link.
you.
Please send
Merry
the link to the
Christmas and
recorded
Happy
seminar.
New Year. Bye.
Thank
Sorry,
you.
Global,
Bye-bye.
you mentioned that?
No, please send the link to the recorded seminar, Manfred.
Sorry?
Please send the
No.
link to the recorded seminar.
Yeah, great. Good.
I
Excellent.
think you have this recorded seminar, isn't it? You will send it.
No, please send, Manfred, please send to everybody the link to the recorded seminar.
Okay, yes, we will do
Several
that.
of my ophthalmology friends from the West Coast asked me to have it at the recorded seminar.
No, sorry, Rose, clearly. If you go to our website, okay, then you can go to the right side with the new registration for the new webinar, scroll down, and there you will find every webinar which has been done. So there are, I think, 12, even there was one with COVID, 12 plus the next, I think on Tuesday, normally we'll have this link completely, yes? So only tell them, go to IUSG webinar. If you do not find the correct email, you will get some message from me too. And then you easily find these things. And I think that's a really, really great achievement that these people, come on, there's such a lot of time difference. Sorry, I'll say about that one. Yes, good night. That's impossible for all of these people. Look for Australia. That's 2 o'clock in the night now. It's lovely to have this on demand for these people. Yeah, so we are very happy with this situation.
Thank you.
But
yes?
Manfred, if somebody could do what Jose Montoya asked, it would be great, because if somebody could just send us the link by WhatsApp, would be much easier. I'll try to get, but I'm not sure I'll be able to.
Again, the link is
It's
on
easy,
our website.
It's easy, Rubens. It's easy, just entering the IUSG page and web page, and you will find all the recorded webinars.
I send you a message, okay?
Okay, thank you.
Bye. Thank you so much again. Thank you so much.
Pleasure to work with you.
Happy holidays. Bye.
Bye.
Thank you, Manfred.
Bye-bye.
Thanks so much.
Thank you, everyone.